Good morning, good afternoon, and a good evening to everyone present here today. I would like to welcome you all to the second talk of our Life Sciences webinar series this year. My name is Sahil Gulati. I'm the Applications Manager of Life Sciences at Gatan. Now, before I introduce our speaker for today, I would briefly go through the question submission process. You can type in your questions in the question panel of the GoToWebinar task window. We will try to answer all your questions immediately after the talk, depending on the time remaining. If by any chance we do run out of time, then your questions will be taken offline, where the answers to your questions will be sent via emails. With that, I would like to introduce our speaker. I welcome Johannes Elferich, who is currently a postdoctoral research fellow at the UMass Medical School in the laboratory of Nicholas Gregorov. A little bit of background on Johannes. Johannes obtained his Ph.D. in 2015 in the laboratory of Ujjwal Shinde at OHSU, working on pH-mediated activation of mammalian furan. He then joined the laboratory of Eric Gaux at OHSU to study mechano-sensory transduction that is involved in hearing. In 2021, he joined the laboratory of Nicholas Gregorov to work on developing methods and software for high-throughput molecule detection in cryo-electron micrographs to enable visual proteomics. He uses a combination of single particle data acquisition and cryo-electron tomography to study localization, stoichiometry, and structural flexibility of the tip link in stereocilia, which are extracted from the inner ear of mice, furthering our understanding of hearing. The title for today's talk is Imaging Molecules in Their Native Environment, Cryo-electron tomography of protocadherin 15 complexes in mouse stereocilia. And with this, I will leave the podium to Johannes.
Thanks a lot, Sahil. Can everybody hear me fine?
Yes, we can.
Okay, great. The reason you can are these fantastic cells that you have in your inner ear called hair cells. And they are really the part of the hearing machinery that converts the mechanical stimulus, that is sound, into an electrical stimulus that your brain can interpret. So here you can see some beautiful SEM images of the hair bundle of these hair cells. So these hair bundles, as you can see, consist of about 100 individual hairs, which are called stereocilia. And they're always arranged in this, even though there are many different types of hair cells, they always have that arrangement, which we call the hair cell bundle, where they're in a sort of staircase pattern. There are rows of stereocilia of ascending height. And one very important fact to notice is that where a shorter stereocilium sits next to a longer stereocilium, there is a filament that connects the two, and that filament is called the tip link. So how does this really beautiful cellular feature of a hair cell enable hearing, and also the sense of motion and balance? So to guide you through this, here on the right, you can see a model hair cell. And so if you were to patch the cell and measure the current that flows over the plasma membrane, you would see a fairly low current. But as soon as I use a blunt probe like the circle to disrupt this bundle and push it to the side, you will notice that the current increases. And the more the bundle is displaced, the higher the current. And furthermore, this change in the current is very, very responsive to the change in the mechanical state of the bundle. So if I repeatedly push the bundle, you could see I get current spikes that very faithfully represent the mechanical stimulus. And that is really the main function of hair cells, to very precisely convert mechanical stimuli into an electrical signal. What you might notice is, and I'm jumping over decades of research here, is when I push the bundle over more and more, that the tip links that connects the stereocilia becomes longer. And because the tip link presumably is fairly stiff, that exerts tension on the tip link. And we know that it's this tension that opens an ion channel that sits at the bottom of the tip link, which is called the mechanosensory transduction channel. And the way this works, how tension on the tip link is converted into that opening of the channel, is kind of the fundamental question that the field has been trying to answer for many, many years now. So let me introduce you to the proteins that underlie this function. So the tip link itself is made of two proteins, protocatenin 15 on the lower end, on the shorter stereocilium, that's connected to catenin 23 that connects to the longer stereocilium. And at the bottom here is the channel. For a long time, it was unclear which protein actually, or which proteins create the channel. But there's some fairly good evidence right now that the channel pore is mostly formed by this protein called TMC. And there are two isoforms in the mammalian here, TMC1 and 2. But there are also two other proteins called TIMI and LHFPL5, which are also membrane proteins that seem to be involved in some form in the formation of that complex. And then this complex is somehow connected through the actin that fills these stereocilia. And later on in images, you will see these stereocilia really packed full with actin. So the complex presumably somehow is coupled to actin using a combination of Berlin, CYP2, myosin 15A, and more recently there's good evidence that ankyrin is actually the molecule that couples CYP2 to actin. But we as structural biologists, of course, want to go further than this kind of cartoon view of the machinery and actually create atomic detail models of the machinery to really understand how that transduction works. And so this is a rendering that I created for a recent review that kind of summarizes the current state of structural biology for this complex. And it's actually not that much. We kind of know what actin looks like. We kind of know, even though there's no structure of TMC, because it is homologous to another channel family called TMN16, we are pretty certain that it's dimer, where both subunits have their own ion conducting pore. And then really the only structural insight we have to the machinery at the bottom of the tip link is the structure of protocatenin 15 and complex with one of the molecules involved in the complex, LHFPL5, that me and my friend Jing Pang solved a few years ago. But how the lower end of the tip link is coupled to the channel and how these other components like TIMI and CYP2 come into it is still unknown. So to tell you a bit about the protocatenin 15 LHFPL5 structure, because it's really the starting point for the story, let me just give you a quick summary. So protocatenin 15 is a catenin molecule, so it has a large extracellular domain that consists of 11 repeats of a catenin domain. Then there is a domain that was previously unknown what the structure is, that we call EL for extracellular linker domain, followed by a single transmembrane helix and then a C-terminal tail. Whereas LHFPL5 was known to be a transmembrane protein with 4 transmembrane helices. And so when we solved the structure, we could see that protocatenin 15 and LHFPL5 were both dimers, so it was a dimer of dimers. And what was interesting that the EL domain, together with the last catenin domain, EC11, formed this kind of collar right proximal to the membrane, which is kind of propped up by the beta sheet of LHFPL5. And LHFPL5 also kind of organizes the 2 transmembrane helices of protocatenin 15. But of course, from this structure, it was not really clear how this would be coupled to a channel and how tension on it could open up a channel, even though we had some ideas. But something that I found personally very interesting about the structure is that when we looked at 2D class averages, is that the whole protein was highly mobile. And I'm only showing here a quick movie of one of these motions. So this is just a couple of 2D class averages put together. And it seems like there is a joint in the protocatenin 15 extracellular domain that is right here between EC9 and EC10, where the whole quaternion chain can almost bend 90 degrees, which was really fascinating to me. But of course, we had no way of knowing whether these conformational changes are actually found in situ at the actual place of mechano-transduction, or if they're just an artifact of single particle analysis. And I think that's a general crux that applies to most structural biology. But the more pressing problem was that despite substantial efforts, there is no structural data available on any of the other components of the channel. So this really hampers our understanding how mechanical stimulus can open a channel. And when I say substantial efforts, we tried everything. We co-expressed all the components, and they just don't assemble in heterologous cells. And probably the reason is that unlike other sensory proteins, for example, like photorhodopsin, if you express photorhodopsin in a cell and put photons on it, you will get a current. But in the case of the mechano-transduction channel, it so much depends on the peculiar cellular features of the cell, so that we cannot reconstitute that in another cellular system. So what we thought we had to do was to look at the real thing in stereocilia. So how do you do that? So first of all, we were quite lucky that at OHSU there was another lab, the Bart Gillespie lab, that had just figured out how to place stereocilia on a cryo-EM grid. So what you do, and this is still completely amazing to me, is you take one of the organs of the inner ear called the utricle, and the utricle is involved in balance sensation. And what's nice about it is that it kind of has a flat surface where all the stereocilia are pointing out. And if you push that against a wholly carbon film grid, the stereocilia will just stick to that grid, and you can remove the utricle, you can take the grid out of whatever buffer you had it in, blot away the excess buffer, and you're left with this thin film of stereocilia that you then can plunge freeze. And if you were to look at that in a TEM, in a cryo-TEM, this is kind of what you see. This kind of random assortment of stereocilia over a grid film. So what we then can do is basically take high magnification images. And we were mostly interested in the tip of these stereocilia. So wherever one of these tips lined up with a hole in the carbon film, we could take pictures. And the way we took these pictures is we used cryo-electron tomography. And since I'm not sure how much people are familiar with this technique, I will briefly introduce it. So the main idea about tomography is that instead of taking just a single exposure of your feature of interest, you take a series of images where for each image you mechanically tilt the sample differently. So normally you tilt it from about +60 to -60 degrees, and at every tilt interval you take an image, and from that you basically get a movie. And so this is what a movie looks like from a stereocilium. So I hope already from this movie you can appreciate really how densely packed these actin fibers are in these stereocilia. The other thing that might spark in your eye are these black dots that are there. And they're just tiny gold beads that we add to our sample to aid the second step of tomography, where we take each of these images and project them back into a 3D volume to get a volugraphic reconstruction. And for this step, it's very important that we precisely know the angle at which we looked at that sample, and these gold beads help with determining that angle very accurately. So after the procedure, we have an electron density volume of that sample. So the way you would look at such a volume, for example, in single particle reconstruction, is that you take an isodensity surface and you get this nice 3D view. And unfortunately, in tomography, that most of the time doesn't work because the data is just too noisy. So what you normally do is you look at isolated slices to that volume. And so here I'm showing you some slices through the reconstruction of that stereocilium from like the XY direction. So we always define the electron beam direction as the Z, the direction of the tilt axis is Y, and the other dimension is X. And what you might notice that in these different views, the XY looks way clearer than the other two. And this is due to a phenomenon that's really important in tomography, and a major problem is it's the missing wedge. So as you can see here, we cannot really tilt our sample to 90 degrees because then the electron beam would be completely stopped. So we have this kind of wedge where we have no information, and that means that our Fourier space is not completely filled, and we will have missing information and artifacts in some of the directions. So in XY slices, everything looks fine. The membrane looks fine. You can almost see individual actin monomers. In the XZ slices, things also still look fairly okay. You can still nicely see the membrane with a bilayer, and you can beautifully see the packing of actin inside. But in the ZY slice, where we're looking at along the Y direction, which is our tilt axis, you can see there's almost no usable information there. And that is because we're basically right looking into the missing wedge. So this is really important later on when you want to find features, and if they just happen to align with the missing wedge, you basically will miss them. So some technical details about how we got the tomograms for the study is, so all this data was collected on a Teton cryos, either at Janelia or at the Pacific Northwest Cryo-EM Center. Both cryoses were equipped with a bioquantum energy filter and a K3 camera and were operated using serial EM. Technically, the Janelia scope also has a CS corrector, but I don't think that matters. So for tomography, we used a 1.7 angstrom pixel size, maintained the energy filter with a 20 electron volts window, and then collected tomograms with a dosimetric scheme. If you're interested in that, we can talk later about that in the questions, at 3-degree intervals from -60 to +60 degrees. We used a total dose of 120 electrons per angstrom squared. So that's about, so these are 40 exposures, so it's about 3 electrons per angstrom squared per exposure. And we used a defocus of 2 to 3.5 microns. And then for processing we did motion correction, motion core 2, and then used IMOD, which is kind of the gold standard for tomography programs, to reconstruct the tomograms, bind them to a 6.8 angstrom pixel size, and use a SERT-like filter to make the contrast really nice. In some cases, we struggled with the fact that the stereocilia seem to deform quite substantially under the electron beam. And in this case, we used a program called tomo-align to correct some of that motion. And sometimes we also further process the tomograms with a neural net denoiser that's implemented in Topaz. So how would we go about? So we have now these nice tomograms of stereocilia tips, and initially we just looked through the slices, and sometimes we were very excited to see these kind of black lines that were about 50 nanometers long, which is the expected distance for protocriterion 15. But as scopes, we quickly realized that we were basically just looking at random features, and we had no way of knowing if this is really our molecule of interest. So what we had to do was to label it somehow. So in our first attempt, we used a fairly standard immunogold approach. So we raised a polyclonal antibody against the protocriterion 15 extracellular domain. So this is just some immunofluorescence microscopy to kind of convince you that it very specifically binds to protocriterion 15. So the knockout, we see no staining. But what we then saw were these, what we then do is we incubate our stereocilia with this polyclonal antibodies, and then with a secondary antibody that's coupled to also gold nanoparticles. They were a bit smaller than the gold nanoparticles we would use for the alignment. And so then we would often see these giant clusters of these gold nanoparticles appear at the tip of stereocilia, which made us very excited because it meant we could show there's protocation-15 there. But of course, it's really hard to interpret this. So this is kind of the best reconstruction we ever got out of them. And you can kind of see a filament that kind of enters this huge density with gold particles. And on the right is kind of our artistic interpretation of that. So the problem is that the protocation-15 molecule gets surrounded by many different copies of the polyclonal antibodies, which then get bound by multiple copies of the secondary antibodies, which are then labeled to the gold. And that makes it just very hard to interpret what's going on. So we knew we had to find a better labeling agent. And so luckily for us, we stumbled over this paper by Maya et Zubel in the Kornberg lab, where they developed these really nice small gold nanoparticles that could kind of be labeled in a one-to-one stoichiometry to another protein. And at the same time, we had some success raising monoclonal antibodies against protocation-15. And especially one of them, 3097, kind of really looked promising to us. And I will not go through these experiments, but what we realized was that it bound to the third extracellular cation repeat. And the reason why that was very ideal for us is because we thought if we were able to get the antigen binding fragment of this antibody, label it with a gold particle, and it then bound to the third cation repeats, it would be as far away as possible from the membrane, which in this case would be here, which is kind of where the interesting stuff would be happening. And it also would leave the first two repeats alone, which are the ones that are responsible for binding to cation 23. So it would kind of be the best of both worlds. So we were hoping is by synthesizing these reagents, we would basically get images like this. And after a few tries, we were very excited to see on I'm not sure why this picture is not loading. I mean, I had a micrograph here, but you also can see here that on single particle images, we could precisely see density that looked like this. So we could see a long line representing the protocation-15 dimer. So this is a recombinant protein. This is not in stereocilia yet. We could see this Y shape at the top, and then the antigen binding fragments that couple the gold nanoparticles to it. And of course, we then were very eager to try this out in actual stereocilia. And when the first TIL series came in, we were quite ecstatic to see that we could see these black dots kind of travel in pairs together at a reasonable about 50 nanometer distance from the membrane. And that was our first indication that we were really able to label individual protocation-15 molecules and stereocilia and take images of them. And the other thing that immediately jumped out to us is, like in the early cartoons I showed you, it's always assumed that there would be like one copy of the protocation-15 dimer at the tip. But here it was immediately obvious that there were clearly more than one. So when we then looked at slices of some of these tomograms, in a lot of cases we could find these 2 AU gold nanoparticle labels that were consistent with protocation-15 dimers. So here I'm showing you 2 examples. And one example is the protocation-15 density extending from the stereocilia and being perpendicular to the electron beam. So it's not hidden in the missing wedge. And the 2 gold particles are kind of in the same plane. So in one XY slice, we can see the protocation-15 densities together with the 2 gold particles. And the dimensions matched up really well with what we were expecting. And in this example here, protocation-15 is also perpendicular to the electron beam, but the 2 gold particles are kind of above and below it. So in an XY slice, you wouldn't see them. And in kind of the side view, you can see the 2 gold particles, but because of the missing wedge, you cannot see protocation-15 anymore. We also observed some examples where there was only one gold nanoparticle label, but we were first wondering if that meant there were monomers of protocation-15 or it was just incomplete binding. And based off some of the best densities we saw, we think they're actually protocation-15 dimers. So here you can, the density looks definitely like there are 2 chains of protocation-15 in this molecule, and just one of them is bound with a gold nanoparticle. And here again is an example where the gold nanoparticles is kind of in line with the electron beam, so you cannot really see it in the XY slides, but you also again can see two strands indicating that it's in fact a protocation-15 dimer that's probably only bound with one copy of the labeling reagent. So we then did a first tally of all the tomograms we have imaged to kind of get a better understanding of how well this labeling worked. So in total we looked at 532 tomograms, and of these 280 contained stereocilium tip, and in about half of them we could also see the gold nanoparticle label. In some of the tomograms we only had the shaft region of the stereocilium, and in this case only a smaller fraction, about a quarter, contained label. And some of them we have both a stereocilium shaft and a tip next to it, and then again in about half of them we saw a label. We also counted how often we saw 2 gold nanoparticles and 1 nanoparticles, and found it was about a 4-to-1 ratio. And then we kind of speculated under the assumption that the binding of the 2 antibodies is independent of each other. That would mean that the binding efficiency, so the fraction of protocation-15 molecules that are bound by a gold nanoparticle, would be 86%. And that would mean that only in a very, very small fraction we would expect to see none of the protocation-15 protomers to be bound by a nanoparticle. We would miss them completely. Of course, the assumption that the binding is independent is might not be true. It's very possible that it's cooperative, but it gave us some rough idea about how well the binding works. So coming back to the fact that there are multiple protocation-15 at the tips, so we then looked at all the tips that we were able to image in our tomogram, and found that in more than half of them we didn't see any gold nanoparticles, and therefore presumably no protocation-15. There's a couple of reasons why there could be. Most ideally you would expect protocation-15 at every tip, but for one, it could be that we're selecting for the longest stereocilia, which would not be connected to a longer stereocilium and therefore would not have a tip link. But what's more likely is that in the sample preparation we damage tips and therefore we don't see any labeling. And the remaining tips, a small fraction had only a protocation-15 at the shaft next to the tip, but not at the tip itself. And that could be just diffusion between the time that we place the stereocilium on the grid and we freeze it. But of the ones where we saw a tip, about 2/3 had multiple protocation-15 on the tip, and about 1/3 had only one protocation-15 on the tip. And looking at the numbers, they're all over the place. Lots of tips with 2 protocation-15, lots with 3, and actually quite a substantial fraction with more than 5. So that was very exciting for us, because it raised the possibilities that tip links might actually be composed of multiple copies of protocation-15 and Cation-23, which has important implications about the regeneration of tip links after damage. And it also fits with recent findings that there might be actually more than one copy of the channel at the tip of stereocilia. Furthermore, in some of the tomograms that we looked at, we were pleased to see that there was additional density that was extending beyond protocation 15. And in this particular example, it extended about 130 more nanometers and then entered this a lipid vesicle at the top. And this length and the fact that it bounds to protocation 15 is very consistent with the idea that these are cation 23, and that this originally was a tip link, but during the sample preparation, the two stereocilia became separated, and one side had to give, and so the lower end of cation 23 basically was pulled out of the other stereocilium. And again, you can see here that there are in fact multiple copies of protocation 15 and multiple copies of cation 23 that formed this link. We could also see kind of the reverse, where in the shaft of a stereocilium we see a bunch of cation 23 molecules, and then we have a lipid vesicle that contains the corresponding protocation 15. So in this case, when the two stereocilia became separated, basically the protocation 15 side was pulled out. And in these images, what was quite striking is that we do see kind of these strong bends, both in cation 23 here and here, but also on the protocation 15 side, there seems to be a very striking 90-degree bend that precisely matches up with that joint between cation repeats 9 and 10 that we saw in the single particle data. And in only one of our 500 tomograms, we actually found an arrangement where it looked like a tippling was still intact. So we had one stereocilia shaft, one stereocilium tip, and of the multiple protocation 15 molecules at the tip there, one had an extending density that inserted into the longer stereocilium, whereas the other copies were unbound by cation 23. This again is an interesting finding, and it's a bit unclear what this really means. Does that mean that the multiple copies of protocation 15 exist there, but only one of them participates in a tip link, and the other one is just a reservoir for regeneration? Or it might be that, again, this sample was damaged somehow during preparation, only one of the tip links survived. But another very interesting feature we kind of saw further down the two stereocilia, which was another structure like a tip link, like a protocation 15 connected to a cation 23 molecule. And in this case, it looks very consistent with the idea that tip links are generated by motor proteins at the base of cation 23, which kind of pull a tip link complex to the tip of the stereocilium. And quite strikingly, in that example too, the protocation 15 had this 90-degree bend between cation domains 9 and 10, which for me makes it very possible that this bending motion does have a real physiological importance, and that it's important for protocation 15 to adopt the geometries that are required during the trafficking of the tip link to the tip of the stereocilium. Okay, so with that, I hope I have convinced you that cryo-electron tomography together with immuno gold nanoparticles allow the imaging of rare protein complexes. And the mechanosensory transduction complex with protocation 15 is probably one of the rarest protein complexes that exists. So if it works with this, it will work with a lot of different things. And the things you can get from that, you can get extremely accurate location of these proteins. We can basically count the numbers just by counting the gold particles. You can actually see different conformations of these molecules, like we were seeing these bending motions in protocation 15. And hopefully you would be able to get structures out of that by doing subtomogram averaging, where you cut out different copies of that protein, average them together to get a higher signal to noise ratio, which allows you to get actual structures. We weren't successful doing that with this existing data set because even though we collected a lot of tomograms, in the end we were still only left with a few hundred copies of protocolate 15, and for good subtomogram averaging you ideally have thousands or tens of thousands of copies. And furthermore, the protocolate 15 turns out to be highly flexible, so any attempt to average them always resulted in very fussy results because they don't overlay exactly. But what we could find is that data that suggests that tipplings occur with more than one copy at the tip of stereocilia, similar to findings for the channel. So that really goes into the idea whether stereocilia in different hair cells might tune their basically electrical properties by modulating the number of copies of the tip link and the channels. And we also could see that protocation 15 and potentially Cation 23. So we cannot 100% sure that the densities we saw are Cation 23 because that's not what we labeled. But both of them seem to have very localized binding points that might help it to adopt the geometries that are required during trafficking, whereas when it's under tension at the tip, it would be completely straight. But it's clear that the detection of these low abundance in small proteins in cryo-EM, even though it's possible, still remains very challenging and kind of a problem you have to solve on a case-by-case basis, and is a very active area of research. And all these, what always strikes out to me, all these problems I've described with proteins being flexible, we're not being able to see them if they don't align perfectly missing wedge, could all be solved if we just get more data. If we can get 10-fold, 100-fold, 1,000-fold more data, we could presumably make all these problems disappear. But in order for this to happen, there are a bunch of bottlenecks we have to overcome. The first is sample preparation. So we need better ways of creating lots and consistent thin enough samples that we can look at. And one very particular exciting technology right now is focused IMB milling for that purpose. The next one is data collection. So with the data strategy we used here, it takes about 20 minutes to collect one tomogram. And since time on high performance electron microscope is precious, it would be better if we could collect tomograms in a short amount of time. And there are some very interesting recent approaches that kind of cut that time down to only a few minutes by basically keeping the camera on while you're tilting the sample. And lastly, and I think that might actually be one of the major bottlenecks, is the computational side. It's becoming increasingly challenging to, first of all, store all the data we're generating, and then also process it all. And in this study here, basically all the identification was done by me personally looking at stuff and manually annotating and identifying it. But in the future, we need better approaches to automatically annotate molecules. And one very exciting development right now is kind of these machine learning computer vision approaches that seem to be very adept of finding molecules in cryo-EM data. So with that, I would like to thank a couple of people, Eric and his whole lab, for really supporting me. In particular, Sarah Clark, who was involved in raising the antibodies against protocation 15, Jing Pang, who has been working with me on this problem for a long time, and April and Aya, who are some research scientists who are really helpful in dealing with the mice and the dissections, which are very challenging. The Bar Gillespie and Nicholson labs, who basically taught us everything about hearing, and then the PNCC and Janelia centers, where we were able to acquire our data, and then Lauren N. Metzgalis, who taught me tomography, and Maya Zubel, who kind of helped us out in how to generate these gold nanoparticles. If you look at these slides online. I think I had the link on the first slide. I'm going to send it again in the chat. You can find here links to, I think, the most important papers if you want to learn more. And with that, I would be happy to answer any questions.
Beautiful talk, Johannes. Thank you very much. Just a reminder to the audience to please submit your questions by typing into the question panel of GoToWebinar task window, which is shown on the right-hand side of the slide. So due to limited time, several questions will be taken offline. Just to begin with a few questions, Johannes. A lot of heterogeneity in terms of the PCDH15 presence, which again would render single particle less useful there. Now, do you think a faceplate would be useful in enhancing the visualization of these complexes so you can better visualize the ones, especially without the gold nanoparticles?
Excuse me, what would be useful? I didn't catch that.
A faceplate?
Oh, a faceplate. Yes. Actually, the first data that we collected with the faceplate, and I think we and a lot of people are finding that, at least at the moment, the faceplates are more problem than they help you. The contrast, it is a bit better, but not that much better than what you can get with just defocus contrast. But it is a lot of problems to keep the faceplate well aligned and conditioned. That really cuts down in your data collection time. So in the end, we stopped using the faceplate for that purpose. But there are new faceplate developments, like for example the laser-based faceplates, and that might change that calculus. So in the future, that might be the case.
Got it. Thank you very much. There's a question about what receptor binds to CDH23 on the second cilium to make that link, the tip link? And is it possible to add vesicles in the sample which are embedded with those receptors to increase the probability of obtaining a full extended tip link?
So Ctn23 itself has a transmembrane helix that inserts into the stereocilium. So it's not bound to a receptor. It itself is embedded in the membrane. And then inside the membrane, it's presumably bound to some motor proteins that traffic it on the actin. But yeah, we thought about labeling Ctn23 too, and that might also be a thing to do. But yeah, still, I think the main problem is that the tip link still breaks so much during data preparation. So I think for the moment, the most important would be to find a sample preparation that is more gentle to the tip link.
Got it. Okay. We have another question. Are there differences in between longer and shorter hair cells?
Between the stereocilia or the hair cells?
The hair cells.
Yes. I mean, there are a lot of different hair cells. So there, for one, are the vestibular hair cells that are used in balance. We looked at in the structure, and yeah, they have a very different morphology. The stereocilia are much longer, and then in the cochlea, actually, there are inner and outer hair cells, and they do have different lengths depending on which position they are in the cochlea, and that's supposed to be important to which frequency range they can sense. And especially there, it seems to be the case that the number of channels per stereocilium changes with the length. And so it would be really interesting if we were able to basically look at only stereocilia from the base of the cochlea and only stereocilia from the tip of the cochlea to kind of see if the numbers of Ctn23s we can see their changes. But also for that, we would need a better way of preparing the sample.
Got it. All right. Thank you, Johannes. There's another question. Is using the gold nanoparticles to label proteins of interest just limited to extracellular proteins, or it can be used for intracellular proteins as well?
That is I mean, for the straightforward case, it is somewhat limited to extracellular proteins. There have been some experiments with coating these with kind of cationic substances that allow them to be taken up by the cell. But just importantly as getting them into the cell, it's also to be able to wash them away. So you get really specific. The only particles that are left are the ones that are bound to a protein of interest. So my thinking is probably for cytosolic samples, we will need to find a way, basically with markers that are being genetically introduced. So that could be like RNA folded structures that might show up very well on cryo-EM data, or there are some ideas of metal binding proteins that are basically creating high contrast features. So I think there's still a lot of ongoing research, and it's not quite clear what the eventual best approach will be.
All right, thank you, Johannes. There is some interest in knowing what kind of grids that you use for your tomography studies. Was it a gold grid or was it copper?
Turns out, I mean, I tried all of them, and the data is probably a mixture, but the one that in the end consistently gave the best data were C-flat grids. And that is a problem because probably 90% of C-flat grids are broken after that procedure. So in the end, what worked best was tons and tons of C-flat grids and screen them for the few ones that are not broken. That gave the best data.
Got it, thank you. And I remember you did mention the antibody that you had used. Sometimes you only see one antibody bound to one of the monomers. So do you remember how strongly the antibody binds? What is the KD value? Might be the reason maybe better antibodies need to be generated to make sure the heterogeneity is less. Do you think that's a possibility?
Yes, we did characterize these antibodies with basically the instrument is called octet, and so the KD is, I don't remember the exact number, but it's well below nanomolar, and the off rate is we couldn't really precisely measure the off rate because it's so slow. So I do think the antibody is really good. The other things that might be going on, there could be isoforms of corticosteroid 15 that influence the epitope. Another big problem is the air-water interface and sample preparation. So the epitope could denature, or the antibody could denature during sample preparation. So I think, yeah, there are lots of reasons why it could be that we don't get both bindings. I do think the antibody is quite good, but of course, better antibodies will always give you better data. There's no question about that. So,
yeah, definitely. Thank you for answering that question. And for the sake of time, one last question: so apart from continuous tomography to increase the throughput of your tomography data collection, what other technological developments you think would have the biggest impact to increasing the outcome of your study?
When it comes to the collection side, I think a big thing is bigger and faster cameras. So if we have a bigger camera, we can collect a much larger area in the same amount of time. And if the camera is faster, we can use a higher exposure and also collect in less time. And they are, together with some approaches of also using beam image shift to collect a large area at each tilt angle. If we then can basically keep the camera on 100% of the time, the area we can image, and therefore the data we get, will be directly proportional to the surface area of the sensor. So bigger sensors, faster cameras would really help us out.
Yeah, that makes sense. All right. With that, thank you very much, Johannes, for your time. And thank you very much, everyone, for attending the talk today.